Welcome to another video in cell biology. Today we're going over the protein ATP synthase. Proteins start off by being made in the process of translation. Transcription comes from, this is my little illustration of the nucleus in DNA. Transcription occurs when the DNA of the nucleus is transcribed into mRNA. mRNA is translated into amino acid sequences via tRNA. And later on, post-translational modification occurs in the Golgi apparatus in the endoplasmic reticulum, and then finally forms protein. And ultimately, there are different structures, different levels of protein. There's the primary, secondary, tertiary, and the quaternary structure. The primary structure is not makes up the smallest component of all of these subunits in ATP synthase. So your basic amino acid, all your different amino acids linked together come up to make a polypeptide chain. And as you can see here, this is an ATP synthase dimer. This dimer is actually said to form the cristae of the mitochondria and is used to stabilize the ATP synthase process. Since ATP is made significant in a large amount in a very short period of time, about 100 ATP molecules per second, and 3 ATP molecules per rotation of the C subunit, which is, you can do the math, a lot of ATP in 1 second. And these are just basically the fascinating part of the molecule, which is the motor, and it rotates and actually releases ATP by binding ADP and inorganic phosphate at this beta subunit site, which is made of alpha helices. And alpha helices are the secondary structure of protein. So we have the F0 domain, which is this region right here, the C ring, the A6L, the subunit A and B, which are part of this peripheral stalk here that is said to actually keep the ATP synthase structure together and aids with the rotation of this subunit. And then the F1 domain is consisted of the alpha, beta, gamma, epsilon, and the delta, and the OSCP, which is the oligosensitive protein. And that's an important part of the peripheral stalk and movement of the ATP. The genetics of ATP synthase, there are 13 different genes that encode in the mitochondrial DNA that are responsible for making the parts of some of these parts of the ATP synthase, and the rest of the genes, which there are several more, are located in the nuclear DNA. And we're going to go over some of the mutations that are involved with ATP. They're not very, ATP synthase, they're not very common. However, they have been discovered, and they have been focus of research by scientists in recent years. And so we went over this a little bit over on the previous video, missense mutation that leads to a different change in conformation of protein at the end of translation, which would be the leucine to arginine, which would be a completely different amino acid, resulting in a different protein structure and an ATP synthase deficiency. Now, this one we didn't actually go over that much in the last video, which is the microdeletion of the AT region of the ATP6 gene, which is actually said to overlap the ATP8 and the COX-3 gene. And two different things happen when there is a deletion of this ATP6 gene. The COX-3 actually is a section of its mRNA is removed, and actually the stop codon is what's removed. The ATP8, which is codes for this subunit right here, which is the base of this peripheral stalk of the ATP synthase enzyme, the A6L subunit, the mRNA cleavage site is actually removed. So whenever there's a polycistronic formation of mRNA after transcription of the ATP region of this particular gene here, is actually missing. So there's not going to be much amplification, there's not going to be very many mRNAs that produce this particular subunit of ATP synthase, which ultimately leads to another ATP synthase deficiency. And that can actually lead to a phenotype in humans, encephalopathy, and it's only actually been found in 2 cases. So this is a way overview of the ATP synthase enzyme and its function, its structure, and some of the genetics that are involved with forming ATP synthase. Thank you for watching the video, and I hope you have a nice day.